Harvard Medical School researchers have unveiled a powerful new artificial intelligence model called PD-Grapher that promises to change drug discovery. Traditionally, drug development has focused on finding a single molecular target, one protein or pathway, and trying to inhibit or activate it. But many diseases are far more complex. They're driven by networks of genes, proteins, and signaling pathways that interact in ways we still poorly understand. PD-Grapher breaks this mold. It is a graph neural network that models how different genes, proteins, and pathways interconnect in diseased cells. It then predicts which gene or gene combinations, when modulated, are most likely to push a diseased cell back toward a healthy state. Rather than testing one drug at a time, PD-Grapher can suggest single-target therapies or combinations of targets, allowing scientists to reverse disease states, not just suppress symptoms. The tool is offered freely, open access, and was described in Nature Biomedical Engineering on September 9, 2025. It was developed with federal funding and aims to accelerate drug discovery by narrowing the search for what works. Marinka Zitnik, Associate Professor of Biomedical Informatics in the Blavatnik Institute at HMS, said PD-Grapher looks like a master chef, figuring out which ingredients produce the desired dish, rather than randomly sampling many recipes. In early tests, the model has already matched known drug targets, flagged known therapies, and suggested novel ones. It has been tested across datasets spanning multiple cancer types, and is expected to be applicable also to neurodegenerative disorders like Parkinson's and Alzheimer's. The potential impact is enormous. For diseases that are driven by multiple interacting dysfunctions, cancers, many metabolic disorders, possibly especially neurodegenerative conditions, PD-Grapher could allow treatments to be identified more rapidly, reduce the failure rate of drug candidates in clinical trials, enable combination therapies that are more effective, and cut cost. The time savings from narrowing hypotheses could mean that some treatments reach trial stage much sooner. Because the model is open access, academic labs, smaller biotech firms, even research institutions in resource-limited settings, may be able to use it to contribute to drug discovery, which has been dominated by large pharma with massive budgets. Moreover, the ability to suggest combination therapies may help overcome drug resistance and disease heterogeneity, when different cells in the same tumor or disease respond differently. On the same day, another major advance comes from Alzheimer's disease research. Scientists at the University of California, San Diego, UCSD, have published a study in JAMA Network Open showing that blood-based biomarkers of neural injury and inflammation correlate strongly with subjective cognitive decline, SCD, in Hispanic and Latino adults, aged 50 to 86. They studied over 5,700 participants in the study of Latinos, Investigation of Neurocognitive Aging, Sol Inca. They measured levels in plasma of 4 biomarkers: phosphorylated tau 181, PTAU 181, neurofilament light chain, NFL, glial fibrillary acidic protein, GFAP, and amyloid beta, A-beta 42/40. The key findings: higher levels of PTAU 181, NFL, and GFAP were each associated with greater subjective cognitive decline, memory, executive function, etc., while A-beta 42/40 showed no such associations. Subjective cognitive decline is when people report worsening memory or thinking, even before standard cognitive tests show deficits. This watermarks a potential turning point. For many underrepresented populations, diagnosing Alzheimer's disease and related dementias often happens late, when symptoms are already severe. Current diagnostic methods typically rely on PET imaging, cerebrospinal fluid sampling, or neurological tests, methods that are expensive, invasive, and less available in many places. A blood test based on these biomarkers could be far less invasive, more scalable, cheaper, and more acceptable for routine screening or for identifying risk earlier. Particularly for Hispanic, Latino populations, which are projected to have steep increases in Alzheimer's prevalence in coming decades. This could help close the gap in diagnosis and treatment access. Together, these two advances, PD-Grapher and the blood biomarker study, signal a broader shift in health science, moving toward earlier detection, more precise interventions, combination therapies, and tools that lower cost and broaden accessibility. PD-Grapher's model could shift the drug discovery pipeline in several ways. First, by identifying combinations of targets, rather than single protein targets, it may help to address diseases previously considered undruggable, because no single target was sufficient. In cancer, for example, resistance emerges when targeting one pathway. Having multiple simultaneous points could improve durability of treatment response. In neurodegenerative disease, where multiple pathological processes, inflammation, protein aggregation, neuronal injury, co-occur, a tool that predicts which combination of interventions might help could accelerate development of therapies or repurposing of existing compounds. The UCSD biomarker study offers potential for changing not only diagnostics but also early intervention. If subjective cognitive decline correlates with these biomarkers, then individuals could be identified years before severe memory loss, opening windows for lifestyle interventions, risk factor control, e.g. cardiovascular, metabolic, perhaps early pharmacological intervention as soon as those become available. It could also facilitate more inclusive clinical trials by enrolling people earlier, from diverse populations, which might improve the generalizability of trial results. There are challenges, of course. For PD-Grapher, predictions must be validated in vitro and in vivo, in cells, animal models, eventually humans. Drug combinations must be safe, with side effect interactions considered. Even if a combination is predicted to reverse a disease signature in cell models, real biological complexity, organ interactions, immune response, pharmacokinetics could derail translation. There's also regulatory, manufacturing, dosing issues with combination therapies. For the biomarkers work, subjective cognitive decline is self-reported, which can be noisy. Biomarker assays need standardization. Cross-sectional design means we cannot conclude causality or temporal order. Do biomarkers rise before cognitive decline, or do early decline and lifestyle disease processes drive both? There may also be variation across populations in biomarker baselines. Nevertheless, both advances are timely. Global aging means more Alzheimer's cases, growing demand for therapies. Drug discovery is notoriously slow, expensive, and failure-prone. Tools that pick better leads earlier have been needed for a long time. Multi-target therapies are increasingly seen as the frontier in many complex diseases. And biomarker-based early detection is a priority for neurologic disease, where once symptoms appear much damage is already done. In summary, on the 9th of September 2025 we saw a pair of advances that could reshape medicine. An AI tool that helps researchers see therapeutic paths and combinations otherwise too complex or too large in search space to test exhaustively. And new evidence that blood biomarkers can detect early Alzheimer's risk among underrepresented populations. These moves forward may accelerate more effective, earlier, equitable diagnostics and treatments. If the promise of PD-Grapher is fulfilled, drug development cycles could shorten, success rates improve, and treatment options expand. And if diagnostic tests based on these biomarkers become routine, people could know earlier, act sooner, and perhaps prevent or somehow slow the course of Alzheimer's disease. If you want to keep up with the biggest breakthroughs in science and medicine, make sure to subscribe, like this video, and hit the notification bell.